Hello and welcome to everyone. Today we're going to talk about the electron transport chain. So what is electron transport chain? The electron transport chain is a cluster of proteins that transfer electrons through a membrane within mitochondria. This is to form a gradient of protons that drives the creation of adenosine triphosphate or what we call ATP. ATP is used by the cell as the energy for metabolic processes for cellular functions. So where does the electron transport chain occur? So during the process, the proton gradient is created when the protons are pumped from the mitochondrial matrix into the intermembrane space of the cell, which also helps in driving ATP production. The use of proton gradient is referred to as the chemiosmotic mechanism that drives ATP synthesis since it relies on a higher concentration of protons to generate proton motive force. The amount of ATP created is directly proportional to the number of protons that are pumped across the mitochondrial membrane. The electron transport chain involves a series of redox reactions that relies on the protein complexes to transfer electrons from a donor molecule to an acceptor molecule. As a result of these reactions, the proton gradient is produced, enabling mechanical work to be converted into chemical energy, allowing ATP synthesis. The complexes are embedded in the inner mitochondrial membrane called the cristae in eukaryotes. Enclosed by the inner mitochondrial membrane is the matrix, which is where necessary enzymes such as pyruvate dehydrogenase and pyruvate carboxylase are located. The process can also be found in photosynthetic eukaryotes, in the thylakoid membrane of chloroplasts, and in prokaryotes but with modifications. Byproducts from other cycles and processes like the citric acid cycle, amino acid oxidation, and fatty acid oxidation are used in the electron transport chain, as seen in the overall redox reaction. So, the energy is released in an exothermic reaction when electrons are passed through the complexes. So, 3 molecules of ATP are created. Phosphate located in the matrix is imported via the proton gradient, which is used to create more ATP. The process of generating more ATP via the phosphorylation of ADP is referred to as the oxidative phosphorylation since the energy of hydrogen oxygenation is used throughout the electron transport chain. The ATP generated from this reaction go on to power most cellular reactions necessary for life. Steps of the electron transport chain. In the electron transport chain, electrons move along a series of proteins to generate an expulsion type of force to move hydrogen ions or protons across the mitochondrial membrane. The electrons begin their reactions in complex 1, continuing on to complex 2, traverse to complex 3, and to cytochrome C via coenzyme Q, and then finally to complex 4. The complexes themselves are complex structured proteins embedded in the phospholipid membrane. They are combined with metal ions such as iron to help with proton expulsion into the intermembrane space as well as the other functions. The complexes also undergo conformational changes to allow opening for the transmembrane movement of protons. These 4 complexes actively transfer electrons from an organic metabolite such as glucose. When the metabolite breaks down, 2 electrons and a hydrogen ion are released and then picked up by the coenzyme NADH+ to become NADH, releasing a hydrogen ion into the cytosol. The NADH now has 2 electrons passing them onto a mobile molecule ubiquinone Q in the first protein complex. Complex 1 is also known as NADH dehydrogenase. It pumps 4 hydrogen ions from the matrix into the intermembrane space, establishing the proton gradient. In the next protein complex 2 or the succinate dehydrogenase, another electron carrier or coenzyme succinate is oxidized into fumarate, causing FAD or flavin adenine dinucleotide to be reduced to FADH2, the transport molecule. So the FADH2 is then reoxidized, donating electrons to Q, so it will become QH2, while releasing another hydrogen ion into the cytosol. But while complex 2 does not directly contribute to the proton gradient, it serves as another source for electrons. Complex 3, or the cytochrome C reductase, is where the Q cycle takes place. There is an interaction between Q and cytochromes, which are molecules composed of iron, to continue the transfer of electrons. So during the Q cycle, the ubiquinol, or in short QH2, previously produced donates electrons to ISP and cytochrome B, so it will become ubiquinol. So ISP and cytochrome B are proteins that are located in the matrix, then transfer the electron that received from ubiquinol to cytochrome C1. So cytochrome C1 then transfer it to cytochrome C, which moves the electron to the last complex. So take note that unlike ubiquinone Q, the cytochrome C can only carry one electron at a time. So ubiquinone then gets reduced again to QH2, starting the cycle. So in the process, another hydrogen ion is released into the cytosol to further create the proton gradient. So the cytochromes then extend into complex number 4, or cytochrome C oxidase. Electrons are transferred one at a time into the complex from cytochrome C. The electrons, in addition to hydrogen and oxygen, then react to form water in an irreversible reaction. This is the last complex that translocates 4 protons across the membrane to create the proton gradient that develops ATP at the end. So as the proton gradient is established, F1, F0 ATP synthase, sometimes referred to as complex 5, generates the ATP. So the complex is composed of several subunits that bind to the protons released in prior reactions. As the protein rotates, the protons are brought back into the mitochondrial matrix, allowing ADP to bind to free phosphate to produce ATP. For every full turn of the protein, 3 ATP is produced, concluding the electron transport chain. So I hope you learned something for today about the process of electron transport chain. So that's all. Thank you.